All right. And although I'd like to start by saying that although land acknowledgement is not nearly sufficient in addressing the issues in their history, we'd like to start by remembering that the Madison Mycological Society and the UW Arboretum were founded on and operate within the traditional and current homelands of the Ho-Chunk people. Although the US government attempted to remove the Ho-Chunk through coercive treaties and violent relocation campaigns, the nation persisted in fighting for their right to live with and protect their ancestral lands. We recognize and strive to combat the continuing white supremacy and colonialism in the fields of ecology, botany, and mycology, as well as the overwhelming culture of privilege and exclusion in outdoor recreation and education. And with that, I'd like to introduce some of our team on this project. My name is Jessica. My pronouns are she and her. And then we also have Kathleen, who will be giving this exciting presentation to us. I'd also like to recognize Julia Whidden, the new Citizen Science Coordinator at the UW Arboretum, who is waving. We're excited for her to be here as well. So if you have Arboretum questions, we can pop those in the chat and maybe Julia can help us answer some of those as well. And also to say thank you to the rest of the MMS board for helping us with this project. Then one last thing to talk about before we get started talking about fungi is that if you have questions, please feel welcome to add them to the chat, and we'll take a break partway through the presentation and at the end to go through those questions. And again, the meeting's being recorded, so if you need to pop out at any time, feel free. And with that, Kathleen, go ahead and take it away. Teach us about fungi.
Yay, great. Okay, I'm unmuted here right before I start getting too excited. Well, first of all, thank you everyone for showing up. Honestly, I couldn't be more excited to be here talking about fungi. Honestly, I feel a little too excited that I'm like a little shaky right now. So I hope I can calm myself down a bit that you can still understand me. But of course, if you have any questions, put them in the chat. If I'm saying anything too quickly or something, to put it in the chat. I am going to try and move through a lot of information though, so it will be a little superficial, and we'll hopefully have time for enough questions that we can get to a lot of things. But as Jessica said, if you have other questions too, put them in the chat. And if we can't keep it contained to this meeting, we'll post things on the website and we'll try and get back to those as best we can. All right. Also kind of making another culturally relevant note here. I feel like we had a really big win for justice today, and I want to bring that here in sort of a moment of celebration in that. All right. Okay, so a little bit more specifically in terms of the overview of this meeting, I'm going to talk a little bit about sort of what is a fungus in general. How do we define that term in that group? So we'll talk a little bit about basic fungal biology and ecology a little bit. We'll spend a little bit of time in sort of this like identifying macrofungi, which are sort of larger fungi, where we'll talk specifically about fungal form groups. You're not going to leave this presentation saying, aha, that's Caprinus comatus, I know that species. It's a little less about putting a species name on things as opposed to just recognizing what is a fungus versus something that might not be a fungus. And then sort of we'll finish the third part of this presentation talking a little bit more specifically about this community science project that we're here to present to you, and how you can get involved in that, what that entails, what you'll need, et cetera, et cetera. Essentially how to take good pictures, is what we'll talk a lot about, and then how to upload those good pictures so that they can be used as scientific information. So let's start here with a little bit of background, right? Like, what is a fungus? That's what we're here for, this Madison Mycological Society that's revolved around fungi. But what are fungi? First and foremost, fungi are not plants. And to some of you, that might seem a little crazy that I'm starting it this way by talking about what they're not, and even referring to the fact that they're not plants. But if we were having this conversation, say even 50 years ago or something, I may be presenting about fungi and talking about them as seedless plants, similar to ferns and mosses, liverworts, basically plants that reproduce via spores, little small propagules that aren't seeds. But they're not plants. And in fact, they couldn't be much more different than plants. In fact, they're actually much more similar to you as an animal than they are to plants altogether. So as you can see in this little diagram here I have called a phylogenetic tree, if you read that from the bottom through the top is moving through time. So at the bottom, that's a really long time ago, and the top is more recent than a really long time ago. You can actually see where that red circle is, that animals and fungi share a much more recent common ancestor in time than either one do with plants. So they're actually much more closely related than we are with any of the plants too. So no offense to the lilacs, but they're a much more distant cousin than all these fungi I'm going to be showing you here. So coming back to this question here about what is a fungus, one way that fungi can be described is that they're a group of organisms that are characterized by having similar DNA sequences, right? So we can tell based on their DNA that they're all quite related, and then we can sort of draw boundaries around that group that makes sense in a way to say, okay, everything in here is fungi, and everything that's less related and outside of here is something else. Currently, using DNA evidence, we've currently described about 150,000 species. But now with the inclusion of DNA evidence in a lot of our scientific understanding, our current estimate to how many fungal species are on Earth is about 5.1 million, if not more. So what that shows is a huge disparity here in terms of like our actual understanding of fungi versus like what there is yet to learn, which is really exciting. I mean, almost daunting exciting, right? But it shows that there's a pretty big gap there. And I wouldn't be surprised if that estimate actually continues to increase as we get more resolution. So fungi are also a group of organisms that don't make their own food. They have chitin in their cell walls, or they tend to. I guess I should put this caveat first, right? I'm going to list a few things here. This doesn't apply to all fungal species, but sort of in general. So I mentioned chitin in their cell walls. Chitin is a really tough polymer. It's actually the same thing that's in the shells of beetles. So if you were to step on one, that's why they crunch so loud, is they also have chitin in there. And I put check marks next to those two, because those are two of the pieces of evidence that before we had DNA evidence, we actually used that to sort of suspect that actually fungi have a lot more in common with animals than they actually do with plants based on some of these things. Furthermore, fungi are also a group of organisms that tend to have a filamentous body form, at least at some point in their life cycle or some part of their overall structure. And they also have a relatively unique lifestyle in comparison, unique life cycle, and lifestyle, I guess, in comparison to animals and plants. And one of those features being that they reproduce by spores, which are these tiny units that are used for widespread dispersal and reproduction. They look something like this under a microscope. And if you've ever heard someone say like, oh, you're breathing in mold or you're breathing something like that, like they're usually referring to the fact that you're breathing in either the spores or asexual spores of a particular species. So they're super tiny and they're really ubiquitous. We're all breathing them in right now, not to make you all uncomfortable. So fungi come in a lot of different forms. I guess, sorry, before I move on with that slide, I do want to highlight that what I was talking about on the previous slide is we can define fungi as being this group of organisms that are closely related, and we can define fungi as having these boundaries based on DNA evidence, but we can also sort of define them based on some of these morphological or body form characters as well as physiological characters, but that this is not an exhaustive list of all of these for sure. All right. So fungi they come in a lot of different forms, and some of these might be a little bit more familiar to you than others. So say the mold on the strawberries there is maybe something you've seen as you found something you forgot in the back of the refrigerator or something. The one above this here though, right, is our baker's yeast or brewer's yeast in general, that's used in all of the baked goods that you love, breads that you love, beer, any of those kind of things that you enjoy. This one here being the body form of a particular group of fungi that are causing a lot of the amphibian declines across the globe, the chytrids. And maybe unfortunately you've seen a body form that looks like this in your basement or something as well, right? In the form of black mold. So fungi can actually look really different, which makes them hard to understand. Please excuse my dog who's rudely drinking so loudly during this presentation, but the focus that we're going to be talking about within this presentation, but also in the project, though, is what we call macrofungi. Macro meaning that they're large. You don't necessarily need a microscope to see their overall body form, maybe certain features, but for the most part, you could actually be walking around and with the naked eye, you could see them without any aid. But I did mention to you that one of the characteristics of fungi is that they tend to have this filamentous body form, right? So I'm showing you a picture of that here. And we tend to use the word hyphae to describe that. So in this top part here, you can see there are these long filamentous strands, basically these long sort of really elongated cells that sometimes have little cross walls in them. So it's a series of multiple cells. And we call that hyphae. But if you're talking about an actual collection of these in a way, we call that a mycelium. And I point this out because there's even though it's a semantic issue, we use that for a particular reason. Because hyphae in and of themselves are extremely small. As you can see, these are microscopic images here. But this mycelium on the other side can actually be, overall, it can be quite large. So this is a relatively small description of one, but in each of those threads that you can see, there's probably hundreds to thousands of actual hyphal strands there. And this overall part of the body form is really important because this is what fungi use for feeding. And they use it in a lot of different ways for feeding. For example, if we refer to that strawberries picture that maybe we can all relate to a little bit, the fungi is actually in there, the hyphae is permeating through that strawberry, releasing all of these water-soluble enzymes, breaking the strawberry down and then reabsorbing it into that fungal hyphae, into the fungal body. And that's how they get their nutrition. But like I said, oops, it's used for feeding. Like I said, mycelium can actually be really huge, which is why this semantic differentiation comes into play. And I'm talking so huge that it can be 3 to 4 square miles large. So if you see this top picture here, you can see this is it's a tree parasite. So you can see actually the line where it's killed all the trees here. But that entire area is approximately 4 square miles large, right? It's estimated to be 2,000 to 8,000 years old and to weigh hundreds of tons. So this particular mycelium actually gets 3 gold medals here for being the largest, oldest, and heaviest of all the organisms. Well, I say of almost all of the organisms that exist on Earth. And so what they actually did is they found out this was all one organism because they went up to the actual mushrooms, which I have pictured here, and they could do genetic testing on those to determine that they're from the exact same individual, to determine that overall thing is actually one individual. But what you might be wondering, because here I'm talking about a mycelium, I'm showing you a picture of this large thing that's a mycelium, but now I'm also talking about mushrooms. So how does this all sort of connect in a way? And I want to use an analogy, which I know can be a little bit dangerous here. So I'll give you the caveat afterwards. But this is very similar to if we were talking about like apples from an apple tree, right? So we have, in terms of the apple tree, we have the actual tree. That's this feeding structure. It has leaves that photosynthesize and this is how it generates carbon and how it feeds. But it also overwinters. It's its overwintering structure. And that's the same thing as the mycelium we were just talking about, whether that be underground or within a log, it's this sort of persistent feeding structure. But in certain times of the year, usually, at least in the case of the apple tree and in the morels that I have pictured here, during certain conditions, when everything's right, it will produce these specialized cute little structures that pop up. And they really only last for a season or for a short while most of the time. And they're really there just to produce, protect, and disperse whatever little dispersal propagules that they have, whether that be seeds or spores. And those dispersal propagules are really the mobile phase that continue this life cycle. So if you think about an apple tree, we're really well versed in that, right? Fall comes around, apples are there, they have seeds inside of them, those seeds produce another apple tree, so on and so forth. But in fungi, I feel like we have a lot less of an intuitive understanding of their life cycle, which is why I use this analogy. So you have this persistent mycelium that's underground and it's feeding. But then during certain times of the year, like right now, exactly, hopefully this next week, right, the conditions will be just right to produce these little morel fruiting bodies, right, that pop up. And within each of those little crevices there, I've included a cross section here, they have little sacs that are producing a ton of spores. So the morels are really just there to produce a bunch of spores, release those into the air, and then they sort of senesce and die as a structure, right? But that mycelium stays around. So I include the big red star and the caveat here because these, in the terms of this analogy, these are similar in terms of their function, but not in their structure, right? A fruit is actually different than an actual fungal fruiting body, which is what we call them. That's one of those holdovers from being part of the botanical world for such a long time, is they're not produced in the same way as a fruit and all of that. The biology behind it is very different, but in terms of their actual function of what's going on here, it does serve as a nice analogy. All right, so I want to get into the ecology here a bit. Let's see where we're at. Okay. So I want to talk a little bit about ecological roles of fungi. So now that you have kind of an understanding of like maybe what a fungus is, or hopefully a little bit more than what you had before, let's talk about what these things do and why they're important. All right, so arguably one of the, if not the most important role of fungi on planet Earth here in a way, is their role as decomposers, or what I have in parentheses there is saprotrophs, meaning sapro, meaning like related to death and decay, and troph, meaning related to nutritional strategy, right? Essentially, they're breaking down dead and decaying matter as a food source for themselves. But it's a really big and important role for the functioning of our Earth in a lot of ways. And so one of these being the recycling of nutrients, right? And I have, for example, here in pictures, I show a white rot fungus and a brown rot fungus. And you may actually recognize these. These are both fungi that can break down woody material, like tree trunks and things. If you've ever been on a hike, right, and you see these rotting logs, and usually they either look like one or the other. They're either this white stringy mess that you can kind of dump your hand into and grab a bunch of that white stringy goodness, or it's this sort of really rich kind of honey amber colored, like a leather colored kind of breaking down of wood, but it's in these little squares, right? It's almost like these perfect little cubes. And if you haven't noticed this, I encourage you when you're out this season to look for that distinction, because once you recognize it, it's so stark and you can see it everywhere. But these are really, really quite important species going on, particularly for the maintenance of forests. So if we would not have particularly white rot fungi, which came around about 280 million years ago, we would not have sustainable forests on planet Earth. We would have likely had one generation of forests where they all grew up and tall, but as they started to die and senesce, they would have eventually fell down. And nothing would be there to be able to degrade that lignin, which is what makes woody material so tough and so hard. There's essentially nothing else on Earth that can degrade lignin, besides from maybe a particular few bacterial species that can do some, but they're not nearly as efficient as white rot fungi. And so without them, we would have essentially had one generation of forests and then nothing after that. So we really can thank them for the fact that we have forests, but we can also thank them for why we no longer produce coal, right? Because once they came around about 280 million years ago, that's when our coal reserves sort of stopped right at about that time. All right, but decomposers I want to highlight here. I showed both woody examples, but they also can break down a lot of other plant material, but it's not just plant material that they can break down. Here we have an example of one that's actually breaking down animal fecal material too. And you can actually do this. I mean, maybe I should be careful what I stress to do at home. You can actually do this at home. If you find like rabbit or deer droppings, you can take them home and incubate them, and you will get a lovely flourish of fungi that actually grow from these things if you keep them hydrated. And there's a whole book, a whole guide on dung dwelling fungi that you see, so you can identify these. I mean, the fun really just doesn't stop. It's so accessible. It's so great. But there's also fungi that can actually break down bone material as well, which those are really, really cool to find if you ever find those in the wild. But I mean, essentially, give it a substrate and fungi can break it down. They're working now with a lot of species too, where fungi can actually break down like cigarette butts and things that most other organisms can't do anything with, or oil, petroleum-based substances too, plastics, and that is what I'm including in that. So it's an exciting frontier in terms of fungi also. Probably, I mean, I guess I shouldn't rank these in terms of what I think are most important, but I think a lot of people could argue that the role as decomposers, but also as these mycorrhizal symbionts, are two really, really majorly important roles that fungi play on planet Earth. So if you break that word apart, mycorrhizal, myco, meaning fungi, which is where the term Mycological Society comes from, but rhizal meaning root, like plant roots underground. And essentially what these are referring to are these really close or symbiotic associations between fungi and plant roots. And I include this little diagram here, mostly just to show you that these relationships can look a number of different ways. This only highlights two, but they can actually look quite different, where the fungi either sort of make this thick mantle around the root and sort of go in between the cells in this apoplastic space, as they call it, or they can actually sort of go into the root and sort of, they don't necessarily puncture the cells, but they push into it as you would push your hand into a water balloon, and they sort of invaginate into the cells to create a lot of surface area. So these can look a lot different. There's all different kinds, but they're really quite important relationships here on planet Earth. In fact, more than 90% of all vascular plant species, or at least families, associate with mycorrhizal fungi. And they're really important in that transition of land or plants moving to land. So we first had these sort of like aquatic algae and things like that, and then plants were able to colonize land, but that really wasn't without the help of mycorrhizal fungi in that way too. So it's a really old, but really important relationship. It's often considered to be mutualistic, meaning that both partners in this benefit, although there is a lot of literature too that really depends on the context that they're in as to whether both partners are actually benefiting from that too. So the benefits to the plant in this situation is that they get much better access to water, as well as nutrients like nitrogen and phosphorus. So essentially these things that fungi are really, really good at doing, fungi are really good at creating a lot of hyphae. They create that large mycelium. So they have a ton of surface area. They can access lots of water, and they're really good at accessing different types of elemental nutrients in that way. Also pathogen protection, really the list could go on. But what do you suppose the benefit to a fungus is from forming a mycorrhizal relationship with a plant? Like what can you assume that a fungus might get from this relationship? And if anyone wants to unmute themselves and chime in here, I would appreciate that.
Food?
I heard food. I couldn't see who said it. Thank you. Yes, I want to give it the sort of long, awkward pause here in terms of looking for a response. Thank you to whoever said that. Yes, that's exactly right here. Let me get back to my screen. Yes, access to a food source. So if you remember at the beginning of this presentation, I said that fungi actually don't make their own food, right? And so in this case, by partnering up with a plant who's really good at creating a lot of carbon-based sugars through photosynthesis, fungi have access to a food, but they also help the plant get access to water, nutrients, and things like this. So they create this really lovely sort of internal economy with themselves. Lichens are another big one that maybe don't get talked about nearly as much. And lichens are also one of these, we tend to think of them as mutualistic relationships, but that has a lot of sort of literature behind it too that suggests otherwise. But they're essentially these really, really interesting composite organisms of at least one fungus, if not more, as we're learning, and some kind of photosynthetic organism, be it a green algae or a cyanobacterium. And they can essentially colonize every terrestrial substrate on Earth, whether it be rock, metal, plastic, ground, tree bark, you name it. They're in aquatic systems, they're at the top of mountains, they're everywhere. They're the first things to move in, usually after a volcanic eruption, whatever. They're really incredible organisms as well. Plant pathogens, some of you may be sort of familiar with this topic in general, maybe fighting them on your garden plants this season as well. But the list could go on here too. I included just a few relevant, especially to this region, like Dutch elm disease, maybe wheat rust, cedar apple rust, which is such a cool one to see. But quite the list here. And in fact, the coffee rust, which is one too, that's exactly why we tend to associate the Ceylon region now with tea. It used to have coffee, and that's exactly why the British drink tea. Actually, if you look that up, there's a whole article about that as to why the British drink tea, and it really boils down to this coffee rust, which is pretty sweet. They can also be pathogens of animals, including humans here. Athlete's foot, ringworm. Ringworm is not a worm. It's actually a fungus growing on your skin that's devouring your keratin, thanking you for all of your nutrients there. Yeast infections and thrush, which is essentially a yeast infection of your mouth and throat. Histoplasmosis, which is really a common thing here in Wisconsin as well as toenail fungus, which is one that's maybe a little bit more common. We're also starting to see too is sort of the emergence of fungi in the world of medicine, right? So in 2019, May of 2019, Denver was the first city in the United States to decriminalize psychoactive mushrooms, particularly psilocybin. Let's see here, I did an updated list here. So following that, Oakland and Santa Cruz in California within June of 2019 and January of 2020 respectively, and then Somerville and Cambridge in Massachusetts in January of 2021. So relatively recently here, we started to see a few other cities. So that's something likely I'll have to continue updating in a presentation like this, always keeping the look, because I imagine that's going to continue to go that direction as well. Finally, some economic uses of fungi. Honestly, once again, this list could literally go on forever, but it starts with breads, baked goods, beer, wine, sake, edible mushrooms, cheese. I mean, it literally just listed almost all of my favorite things right at the top there. So if you like any of the things on this list, you can thank a fungus for that. And if you're not yet impressed, I have a few extra tricks up my sleeve here. Fungi, certain species of fungi can actually trap nematodes, which are like tiny little worm-like organisms that live in soil. And they can do so in such amazing different ways. For example, here I have highlighted, they can actually have these specialized cells that create these little lassos. And when they detect that a nematode has actually gone through there, they inflate those cells and it traps the nematode within that lasso, which is pretty amazing. And finally, there are fungi that can actually glow in the dark, which is what we call bioluminesce. And this may seem like something that only happens in the tropics or somewhere seemingly distant and more magical or something than the Midwest. But I assure you that there's a lot of species actually around here that fall into this category. You just have to find them when they're fresh and get yourself in an extremely dark room. So in fact, on our t-shirts, that's one of the bioluminescent ones that we have there as well. So. All right, let me see what time it is here. This thing blocks my time. Okay, 6:30. I know I can get excited and ramble for a little bit, but okay, taking a moment here, since we're at about 6:30 and we've just gotten through, honestly, a ton of information that I only really just like dipped my fingers into a little bit of it, right? And that's sort of the purpose. We can't talk about everything that's exciting about fungi in 30 minutes because that would just be impossible. There's just honestly way too much to talk about. But hopefully that gave you a little bit of an overview. If you're new to fungi in general, like what is a fungus? What do people mean when we're talking about that? It's much more than just the mushrooms that you're finding in the grocery store that go on your pizza or in your salad or something too, right? But also that they're extremely important. And I only highlighted even just a few ways in which they're important. And I could talk about each one of those probably for a month. So if you're interested in that, keep coming to these meetings and you'll definitely hear more about this. So what I'm going to do is I'm going to talk a little bit about this sort of identifying macrofungi. Now that you've learned a little bit about sort of what fungi are, why they're important, let's show you some pictures of them. So if you're new to this as well, you can at least get sort of a visual idea of just how variable even macrofungi can be. After that, we'll hopefully take, if we have some time, a couple minutes for just like a few questions, and you can type some things in the chat. We can finish some of that at the end. And then we'll talk to you about what the project looks like and how to get involved in that. Okay. All right. So we've titled this the Gallery of Form Groups, because really how we have these grouped is sort of based on what are actually called these form groups in a way. It's fungi that really look the same. In other words, they share a similar morphology. But one thing I want to highlight is that doesn't necessarily mean that they're closely related. So we'll show you some examples of that too. But just because two fungi look very similar, that really doesn't mean they're closely related. But we use these groups to actually make communication more simple and identification more simple of these species. So it makes it much more practical to be able to talk about it if you can have some sort of like anatomical terms to talk about fungi or talk about them in these groups, as opposed to simply saying, you know, talking about something purely in a DNA related context. So once again, as is true with essentially everything in this presentation, this is not an exhaustive list, right? This is just showing you a lot of the cool ways that fungi have evolved to disperse spores. But honestly, this list could go on forever. So if you think this is cool, keep checking them out. So first and foremost, probably the most obvious here, oops, I'm on a roll, okay, are the guild mushrooms, right? This is sort of similar to the ones that you do buy at the grocery store. Like if you've had a portobello mushroom before too, those gills are even more obvious on those. So in this case, the fungus or the fruiting body itself tends to have what's called a stalk. It used to be called a stem, and you can still call it that, but that's definitely one of those botanical holdovers. That's much more of a plant term. So a stalk, some kind of a general cap, just sort of like what you see here. And then in this case, on the underside of that, we have what are called gills, which are these little plate-like fleshy structures. And I should have included a picture here, but if you were to zoom in really close on one of these gills here, what you'd see are these protruding specialized cells that are producing spores on the end of them that drop them off or shoot them off or dangle them off. They do all kinds of different things. But this is essentially when someone is talking about a guild mushroom, or if you use these terms, this is what you're talking about here. So let's compare that to the boletes, right? So once again, we have a stalk, we have a cap, right? We have that sort of traditional, what we consider to be a mushroom shape, which before I move actually too far from the guild mushrooms, I should make the note that the term mushroom in and of itself is is really referring to this type of fruiting body here that has a stalk, a cap, and gills. Mushroom is a very actually particular term in mycology, but we tend to use it to talk about everything, right? So when we're talking about morel mushrooms, we're actually using that word incorrectly. It's a fungal fruiting body, it is, but it doesn't have the stalk, the cap, and the gills, so it's technically not a mushroom. If you like those kind of details, you can hold on to that. So in this case here, back to the boletes, we have a stalk, we have a cap, but if you were to flip this over and look on the underside, as you can sort of see here, what we actually have are pores. And I have a better picture coming up a little bit later in here that you'll be able to see this. But if you were to zoom in on these pores, what it sort of looks like is if someone took a tiny thumbtack or a pin and poked a million holes into the bottom of that surface there. That's what these look like. If you were to cut that open and look at it under a microscope, what you'd find is the same exact cells that I was talking about on those gills, those specialized cells that are producing spores. You'd find the same specialized cells producing spores along the tubes within those pores. All right, the polypores, which maybe some of you have seen some of these as well, these tend to be more of like the perennial group. So these stay around a little more. They're not as transient as some of the others. And once again, they're sort of shaped like a shelf usually. They're kind of coming out sideways from the tree or the substrate or whatever it's growing on, as you can see here, almost like a horse hoof too. But if you were to flip that over or look at the underside, kind of like we can see in this picture, once again, you have pores, which are like tiny little holes. And that's where this name comes from, polypore, poly meaning many, pores meaning, you know, these small pore-like openings. So we have the polypores. But once again, you know, I started this form group by saying that we have these groups and they look the same, but they're not necessarily related, right? And here's a pretty good example here, right? So from the top, as you can see on the left here, right, it looks like a polypore. It's coming out sort of shelf-like from the substrate, right? And you would walk over to that almost for sure expecting you're going to flip this over and potentially see pores. But in fact, you actually see gills on this one, right? So this is a perfect example about how these form groups work, but they're not necessarily tied to the relationships of these species, and there is some variability within there as well. All right, a couple interesting ones that aren't as common, so maybe you haven't seen these one before. Toothed mushrooms, the ear pick fungus here on the left, which is such a cute little name for this. I think this is one is just magical. I mean, look at these little teeth on the bottom here. That's definitely where it gets these names from. But essentially what it has is, instead of pores, where it's these sort of holes and the tubes are essentially openings, these are sort of actual discrete tubes that are coming out of the undersurface that have those specialized cells within inside of each little tube here. And this one here on the right, if you're familiar with that one, the hedgehog mushroom, does grow around here, which I haven't had tons of luck since moving to Madison finding this, but that's a super tasty one here, very cute and very tasty. Clubs and corals. So some of you may have seen these as well. They're a pretty diverse group too, and they can have a lot of cool colors in them also. They can be these big, relatively large club-shaped ones, which are sort of like larger than your finger in a way. And then some of these more like delicate ones that look a little more like undersea corals that you should see like little sea monkeys swimming around or something like that. Jelly fungi, oh, these are such a good group. And honestly, I've seen a lot of these around too, and they were hydrated once we were getting all that rain. So they were really, really lovely. So these ones, like the name implies, is they're sort of gelatinous, but when they're dried up, you would never guess it. And sometimes they're almost like imperceptible when they're dried. They just look like a little flat, shiny, kind of blackish brown smudge on bark. Or if it was the orange one, it just, it's almost so minute, but then once they hydrate, they become huge and just really lovely and voluptuous. They're just really, really wonderful. But that's another example, clearly very different than the gilled fungi, gilled mushrooms that we started with when we first started looking at these. All right, a big group here that includes like the puffballs, the earth balls, earth stars, and stinkhorns. And these are all included on one slide, even though they actually look kind of different, but these all used to be considered in this gastroid fungi. And actually they technically are in terms of the form groups here. They call them gastroid fungi, meaning they're like stomach-like fungi because they produce their spores internally. But you can see that they actually are very different. We used to consider these a group, like an actual related group. But now due to DNA evidence, we know that they're actually pretty different as well. So in this case, we have like puffballs, Whereas when, if you see these when they dry out, they'll develop a little hole in the top and they can actually shoot spores quite a long way. And these puffballs can be really large here, as you can see with our friend Alden, who now lives in Michigan, but he was the founding president of the Madison Mycological Society. You can see that these get quite large as well. The stinkhorns are always a really interesting ones. They pop up on UW campus all the time if you're ever looking for them in the summer. And it's always a fun time when you see these things popping up in the middle of a college campus and you have students who have never seen fungi that look like this before. It causes a ruckus, I know, amongst undergraduates. Truffles are another interesting group, which are these sort of like hardened fungal masses that live underground. And they're really lovely. They often smell really great and taste really great if you've had any truffle products. And we actually do have truffles in the Midwest as well. Hopefully I'll find more this year and I can show you more in our monthly meetings as we have them. But I found quite a few last year, which was really wonderful. Cup fungi, which any of you have been out, you may have seen some of these red Sarcosypha cups out there, or maybe some of the devil's urn, which sort of fall into this cup category as well. But these can be super tiny, like these little yellow ones here, or actually relatively large, like we see in the scarlet cup over here as well. And a little bonus here as well, that this still is, it sort of falls in that weird boundary of being a macro fungus, but also having, you essentially need a microscope to be able to identify these as well, is crust fungi. And this is a really, really variable group that just is all over the place. And sometimes, it can even be hard for a trained mycologist to look at something on a stick and say that's a crust fungus versus it's potentially something else. So a really variable group, but actually a lot of cool diversity hidden in that group as well. So. All right, we're at about 6:42, which I think is, I want to leave time for questions, and I know we're probably getting there a little bit. How about we take like 2 to 3 minutes or so to answer a couple questions that folks might have now, if that seems appropriate. Jessica, does that sound good to you?
Sure. So we had one question in the chat that asked, where is the humongous fungus located?
Oh, good question. Yeah, I don't think I mentioned that. I don't know if it was on that slide at all. So that big humongous fungus that I showed you, that's actually located in Malheur National, is it National Forest? No, Malheur Field Station. It's out in Oregon is where it's at. Yeah, so that's actually within the United States, which is pretty sweet.
So Kathleen, another question all these are really good questions. Do mushrooms grow in prairies or only in woodlands?
Oh, that's a great question. Yeah. And, honestly, I should probably be a little cognizant of that too, because I feel like when we talk about these, right, like I'm showing you pictures of them breaking down big woody substrates or them associating with tree roots. That was what the images are. And that may have been misleading. Actually, prairies can boast a ton of fungi. They actually can have a lot of macrofungi like I'm talking about here, like morels will actually be found in prairies. One of the times I found the most prairies in my life was actually after a prairie burn, when all of these little morels were on essentially fire. They were smoking, which they were like the best tasting morels I've ever had, I will add. But no, there's lots of fungi in prairies, but oftentimes if they're macrofungi, you just don't see them because the grass is so dense, and you're also going to have less of those macrofungi in prairies. But if you start talking about more microfungi, like what are called rusts, which are these really bright orange fungi that can essentially they're pathogens on plants and things like that. Oh, you'll find a ton out there. So you just have to kind of come in with a different lens. You're not going to look down and find like a giant gilled mushroom. You just won't be able to see it in the grass. But once you start sort of attuning your eyes to different types of fungi and know what you're looking for, there's lots there. A lot of endophytes that live within plants too, but those are harder to study and see.
And we'll do one more question. Are crust fungi a type of mold?
That's a good question. I technically no. And I should probably look up what constitutes that mold definition in general. I think what I've gathered in terms of like what a mold is it usually tends to be these fungi that fall in the group of, not always, but sometimes like the zygomycetes, where they have this really loose filamentous body form, and they tend to grow on some of the substrates that we interact with in a way. I don't know if that's like a perfectly great answer to that in general, but crust fungi tend to be a bit different. They're sort of in a different group than usually what we consider to be the molds, like a relatedness group in that way. And they also just tend to have a totally different and more in some ways complex structure. It's less of this like hyphae just free floating through a substrate, as they have these well-defined characteristics. Like, so in this one here, you can see there's little teeth on here. It has this sort of defined margin. They have just a little bit more structure to them than usually what we consider the growth form of a mold.
Thanks, Kathleen. We have a few more questions, but I think we'll save them till the end so that we can start chatting about the project.
You bet. Yes, okay. All right, so a bit about introducing you here to the project. Before I get into this, so essentially the kind of the goal of our project here, this community science project that we're here to talk to you about, is that we really want to play a better role in helping to document fungi, right? And we've been doing this for a while at the UW Arboretum in particular, but this can also be done within Dane County as well. And so why is it important to even do this kind of work? Why should we care? Well, as I highlighted at the beginning, right, is there's sort of this like complete disparity between what we know about fungal species versus how many fungal species exist. So we're not going to be doing necessarily a lot of the work to differentiate one species from another with this project here, but it does become important to study fungi in a little bit more of a way, given that there's just so much that we don't know. And having said that, you put that in the context of a changing globe, right? With a changing environment that we don't necessarily know how things are quite going to respond to, particularly for an understudied group like fungi, right? That haven't been sort of well documented since the 1800s, like we have with plants and animals and things. Fungi, we just don't have those kind of records for, as well as we do have other taxonomic groups. So kind of trying to get a better understanding of who is where in this impending response, or this sort of impending change that we have going on, is really actually quite important. Also, fungal fruiting bodies can often be so transient and actually really hard to find, right? So some of these will be around, they're perennial, and so they'll be there all year. Some of them literally pop up in a day in mulch beds, and then they deliquesce or they senesce away, and you don't see them until next year at that same time. So it makes it really hard for researchers to also have a good understanding of like what is where all the time, and to be tracking when they're fruiting and seeing if there's changes in those fruiting too. So that's why it's really, really important to have a ton of set of eyes over a large area that are looking at these things and documenting these also. What do you need to participate for this project? So ideally, you'd need a smartphone, just because it has access to the internet and your phone all in one. It makes things easier, but a camera and access to a computer also work just easily as well. And the final, but also the most important requirement here is a feeling of excitement about fungi. So if you don't quite have that yet, I hope once you go out and start looking that will find you before you find it. A little bit more explicitly about what this project sort of looks like. What we're trying to do is we're trying to document this fungal diversity through photography and observational data. And we're going to be utilizing the platform iNaturalist, if you've heard that, to sort of help combine those efforts together. So what you would do to participate in this project is you would create an account with iNaturalist, which is completely free. And if I can set it up, that means anyone can set it up. So I shouldn't say that, but I'm not always the best at troubleshooting techie skills. And it's relatively straightforward to do this, but we are very, very happy to help you if you have any questions along the way. And then what you'll do is you'll go out and about and take photos of fungal specimens. We've done a lot of work at the UW Arboretum, and so that's a great place to go that's in town, that has a lot of different habitat types, and that's relatively, well managed and taken care of. So you're likely to see more diversity there than you might within the Madison settings or anywhere else in Madison proper. We'll talk a little bit about how to take a good photo, and then we'll explain the two projects as well. So what you'll do is you record some of the habitat and observational information that you see to support these observations. You'll upload those into iNaturalist, which combines those together. And then what you can do through that portal is you can see what other people are looking at as well. So I'm going to go through this, and to be completely honest, it's 6:50 and I forgot how many slides I had put in here for this. So this shouldn't take me too long to get through, but if I go a little too quickly, put some questions in the chat and let me know what kind of questions I didn't cover. All right, so in terms of the type of photos, like how is it that you take a good picture of fungi? What constitutes a good picture? And we have a lot of really helpful resources on the website actually that cover this a lot more in depth. So for example, though, I have these general image categories that you might want to target. So let's say you're out in the woods and all of a sudden you see what you think is a fungal fruiting body on a stick, as we can kind of see right here, right? Oop, that's what we think it is. So what you want to do is you first want to take a picture that's unaltered, just completely in situ or in situation, as we call it, right? You don't want to manicure it too much. Just take a picture just like this, because this actually conveys a lot of information for us. One thing that would have been ideal here for me to do that I didn't do is to include some kind of a scale bar, like a pen or a ruler or even my thumb, so that we can kind of get an idea of how large this fruiting body is. The next sort of general image category that you want to think about is then taking a picture that is a little more altered, so we can actually see some of these features a little bit better. So what I've done is, you can see I just removed the twigs out of the way. Again, if I had used a scale bar or something, that would have been really useful, right? Is this mushroom cap, is that 2 inches big or is that 2 centimeters big, right? And I don't have anything in this particular photo that tells us that. I've zoomed in a bit here more in terms of this image category 3 to actually get the features of the top of the fruiting body. So you can see here, we can see the colors a little bit better. And you can actually, from this photograph, you can see here that this cap actually has little hairs that grow along the edges of this cap. And it turns out that's actually really important for this particular species identity. Note, you don't just have to take one picture for each category. You can do as many as you want. I'm just highlighting general categories that, like, at least one picture from each of these categories is really good to take. Image category 4, the underside of the fruiting body. So if you're familiar at all, if you're in any of these like social media fungal identification groups, you'll find that this is the one that often gets left out. People will post a picture of the top of the fruiting body and no one will identify it for you because they say, we can't see the underside. We don't know if that thing has gills or if it has pores or what. And so for this one, for example, you may have looked at that and you may have seen the top cap and think, that has gills underneath. But this one actually, surprisingly, has pores. As you can see on this structure here, these tiny little circles. So you want to take at least one picture of the underside. And this is sort of what I'm constituting as my scale bar. And these is the tip of my finger. So you can get kind of an idea of the size of that fruiting body. In terms of image category 5, you want to take a picture of the side of the fruiting body. That way you can sort of see, does this thing have a stalk? And in this case, yes, it does. How tall is that stalk relative to the size of the cap, right? Some of these things that might be really important for determining what the species is from something else. Once again, a scale bar is also very, very useful in these. And this image category 6 here is additional information and features. So anything else that you may have noticed that seems worthy of documenting, right? At least when this is coming in pictures, it's better to have too many pictures of something than not enough, right? So I'd say go for it. In this case, and some of you may have noticed that in the first pictures, there were actually other individuals of sort of different developmental stages within here as well. So even though they were smaller, they actually had darker caps. And it turns out these are all from the same species, they just look different. But that's useful information to document in case, in terms of an identification, does the cap start at this really sort of like light honey brown and then age to a dark brown, right? If you only had the one, it's hard to know. So if you can document some of that supporting contextual information, that's really useful. So if you're unsure if something's unique, photograph it. That's really useful. And if you want to, I put the link in there. If you want to see how some of the features that are captured in these photos go along with that identification, you can visit that link. Here's another example here I'll go through quickly. So once again, we have this sort of unaltered in situ situation here. I think I see a fungus in there. I've manicured it a little bit so that you can see it a little bit better. And I've taken a picture of the top of the fruiting body, right? In this case, it doesn't have a cap, but I've taken a picture of the direct top of the fruiting body so you can see what that looks like. I've taken a picture of, in this case, the underside, which is sort of the same as the side, but you can see it has that scaly pattern on the side, which is useful in terms of identifying this species. And I did actually collect this one. And when I did, I noticed that it has this fuzzy stuff kind of towards the bottom. And I thought, well, that's weird. Maybe that's typical of this species, maybe not. I should photograph it. And it turns out it actually is useful for this species. Here's a nice shot of the side of the fruiting body. And once again, some of this contextual information. So I ended up seeing another patch and saw that not only do they grow sort of solitary, but they can be found growing in clusters. And I also found this one here that has more of a really kind of honey colored brown interior than that really dark color as well. So I thought I'd capture all of that as well so that I have sort of this nice gestalt understanding of what this species looks like. And if some of you may have seen this one already this year, this is Devil's Urn. And you can once again visit this link here if you want to compare some of the features in these pictures with what's in that overall description. In terms of some field notes, and probably referring to the documents that we have would be good for this too, since there can be kind of a checklist. But images themselves, like I say here, they can be worth 1,000 words, right? So those are sort of the meat of your observations in a way, but a few supporting notes can actually go a long way as well too. And you can write these down or you can add them directly into your iNaturalist observation. But the one piece of advice I'd say is don't assume that you will remember this when you get home, right? Do it right there when you have it. Because I can't tell you how many times I got home and I'm like, oh, I'll never forget the composition of that woodland I was in. And I get home and I can't remember what was there or what was happening when I collected that. So for example, in this picture here, which was one of the first ones that I showed you, if that particular leaf hadn't been present in that picture, right? So maybe we didn't know that an oak was present, which actually this species tends to grow on the wood of oaks, right? If we didn't have that leaf in that picture, we might not know whether oaks were present or not in that system. So we lose a sort of piece of evidence in terms of our identification of this one. So I've listed a few things that are really common in terms of taking field notes, such as the date and location you were at, maybe what substrate it was going on, even if you can just say it was a deciduous tree or a coniferous tree like a pine or something, some general habitat notes, as well as some other things. And there's also some extra credit ones here if you want to take a stab at the form group, even go a little further with some cat morphology and get more into the mycological side of things. We encourage you to do so. All right. Jessica, I'm going to pass things off to you, and I apologize that I kind of overestimated how much time I was taking here, so please forgive me.
That was great, Kathleen. Like you said, using iNaturalist in this project is really simple, so it won't take me very long to explain or show all of you any of this. But I can't advance the slides, so I'm going to have to ask you to go to the next slide for me, Kathleen. So just to reintroduce iNaturalist, we've been talking a lot about how this is the tool we're using to document these fungal fruiting bodies. I just want to give you a tiny bit of background so that you know what the platform is. There's a mobile app and a website that you could use to do this, but iNaturalist is essentially this big network of naturalists, of citizen scientists, of biologists, and it's used worldwide to sort of document observations of nature. There have been over 65 million observations as of this year, and it provides really valuable data and information to researchers, to land managers, to organizations. So it's a really exciting way to look at what's happening and document observations. And so the first step in joining this project is to create your account on iNaturalist, which you can do very easily. You can move to the next slide, Kathleen. So to join the project in Dane County is really easy. Any observation that you upload, if you're in Dane County, will automatically get added to that project as long as you have an account on iNaturalist. And you can follow the link here to look at that project and see what other folks are documenting. Next. To join the Arboretum project, you can follow this link. You'll get to a website that looks like this, and then you click the join button in the top corner. And so this Arboretum project is exciting because there are some potential for some other components of the project. If you're part of this group, there will be the opportunity to sort of connect and interact with this community and see what folks are seeing. There will be opportunities to be more involved with the project, to potentially collect some of your specimens and have those be added to our library of fungal specimens and have their DNA be sequenced, and the potential to attend some collection and identification workshops in the future. So I'd recommend that you participate in the project at the Arboretum to help us figure out what fungal diversity is going on there. But also just look at fungi and document them and photograph them anywhere you see them would be my recommendation. Then I'll just walk you through how to upload observations. This is a screenshot of what you would see on the app. So if you open the iNaturalist app, in the corner, you would see a little plus button, and you would go ahead and click that once you have found a fungi that you want to take a photo of. So you could either have a photo that you've taken already on your phone and upload it in this way, or you can find a fungal fruiting body, whip out your phone and get ready to snap a photo in this way as well. So you press the plus button, you'll get taken to the next screen, which will look something like this. So if you uploaded this photo of these beautiful reddish colored fungi, the first thing that you'll see, you'll have, you can press what did you see? And iNaturalist will give you some recommendations of what you may be seeing based on your photos. So the more photos you have, the better the suggestions will be. And so you can go ahead and choose one of those observations, one of those choices. Once you've chosen what you think your best guess on identification, you want to put those other observational pieces of information into the notes. Make sure that you have the date, and make sure that your location is enabled on your phone, because we really want to know where we're seeing these fungi. And then all you have to do is press the little check mark, and your observation, if you've joined the group, will automatically be added to the project. All right, so it is really very simple to add observations. I'd encourage folks to check our social media accounts in the iNaturalist group for frequent challenges. We'll send you out to look for certain things that might be popping up during particular points of the season, or that might be really interesting, or that might help you learn more about fungi. So look for those challenges and updates about the project as well. And you can follow those two links to our social media accounts. One other thing that we'd like to note is to please be respectful of the land, of the trails, and of other people you might encounter on this project. We'd encourage you to use good practices and be kind, do things like social distancing, make sure that you're taking care of yourself and others and the land. Another thing is that we may contact you at some point if you've joined the UW Arboretum project about collecting a specimen with us. But if we don't, please don't take any fungi from the Arboretum. There are certain places in Dane County that you are allowed to collect fungi, but the Arboretum isn't one of them. So just take photos and document that way. And at some point, there may be some opportunity to collect. And then just a few final notes to end on. There are guides to all of these things that Kathleen and I have talked about about how to take good photos, about the morphology of fungi, and those are available on the MMS website. You can download them. We might occasionally share images that we really like that have been posted on the iNaturalist projects on our social media. And if you prefer not to have us share your photos, please send us an email. But we're really excited about engaging with you all on social media and sharing what we see and getting excited about what you see. And then any questions, you can, again, check the website for resources or contact us. And so again, thank you. We're really excited to learn alongside you. I want to remind everybody that the presentation will be recorded and added to our website so you can access it later. And then one other thing I'd like to ask of all of you is to please fill out an evaluation form that I'll drop in the chat. That will help us make the project better, help it make it work better for you, and help us create a better community through this project.